Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by AGTC, Allergan and Abbvie Company and Novartis. We'd also like to thank Accessible Media Inc., our national media partner. Everyone here should download the AMI app, which is a fabulous resource. And you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below.
So we have three wonderful guests today who are going to talk about different topics, all related to AMD. So just to give you a quick rundown, we're first going to speak with Dr. Peter Kertes about dry AMD and geographic atrophy. Then we'll be joined by Dr. Levi Blazer, who's going to speak about a newly funded, FBC-funded research project. And then finally, we're going to speak with Dr. Bernie Hurley, who's going to share with us some information about biosimilars, what those are and what impact they may have for AMD patients. So thank you to all of our speakers for joining us today. And I think we'll get started right away with Dr. Kertes. Dr. Kertes, thank you so much for being here.
Thank you, Morgan. Thanks for the invitation. It's a pleasure to be here. With you.
Yeah, absolutely. So maybe just to start off, could you maybe introduce yourself, provide a little bit of background on your work with AMD?
Sure. My name is Peter Kertes. I'm a retina specialist. I do both medical and surgical retina, and of course, a big part of any retina specialist practice is macular degeneration. I work at the Sunnybrook Health Sciences Center, which is a university-affiliated hospital in Toronto, and very happy to be here to talk to you about dry age-related macular degeneration, or in particular geographic atrophy.
So just to give us a bit of context, we've heard these terms, wet AMD, dry AMD. Could you tell us a little bit about what these two different types are and how they might be different?
Sure. We divide macular degeneration kind of broadly into a dry and a wet form. And what makes the wet form of macular degeneration wet is the growth of abnormal blood vessels. So what can happen in macular degeneration, remember the macula is the central portion of the retina. So it's the portion of the retina that's responsible for your central vision, your reading vision, your driving vision, your color vision. And what happens sometimes is that abnormal blood vessels can grow underneath the central part of that retina, and it can leak fluid and cause swelling, and can cause vision loss. And it's the wet form of macular degeneration that's responsible for the lion's share of vision loss in macular degeneration. The dry form of macular degeneration is really everything else, and it has a huge range. It could range from very mild and minimal symptoms to very severe forms of vision loss. So you don't need to have these abnormal blood vessels growing to have significant vision loss. Do you want me to just go on in talking about geographic atrophy, Morgan?
No, I think that's good to start. Can you have both conditions at the same time? Can you have dry and wet? For
sure. I mean, even the most severe forms of wet macular degeneration start as dry macular degeneration, and virtually everybody beyond a certain age will have some kind of the sort of wear and tear changes that we see in dry macular degeneration. So you start with dry, and on occasion, and sometimes it can convert into the wet form of macular degeneration. A more common scenario is someone may have wet macular degeneration in one eye and dry macular degeneration in the other eye. And of course, that eye with the dry form of macular degeneration is certainly at risk of progression or transformation into the wet form.
And what do you recommend there for people that have the dry, just to continue to monitor it, see your eye doctor regularly? Is that sort of the best course?
Yeah, for sure. So, we always we always want people to check their vision. And, when we say check your vision, we want you to check it with one eye at a time. So you want to cover one eye and check the fellow eye. Sometimes we see people relatively late in the course of disease because, they've, with both eyes open, they may not have noticed that they've lost vision in one eye. So it's useful to check vision at one eye separately. And of course, regular eye exams with your eye care professional is worthwhile as well. For those people with what we would classify as high risk dry macular degeneration. So these are people that we know are at higher risk for developing the wet form of macular degeneration or the more severe forms of dry macular degeneration. We usually recommend, there's a concoction of vitamins that we recommend that people take to try and help prevent vision loss. And then there's lifestyle things that can be used to help prevent vision loss and macular degeneration. We know that people who eat lots of fish tend to get, less macular degeneration than people who don't. We know that a diet that's rich in green leafy vegetables is helpful in preventing vision loss from macular degeneration as well. And we know that smoking is probably the most important controllable risk factor. It certainly puts you at risk for vision loss and progression in macular degeneration. So for those that smoke, we recommend that you do your best to try and at least cut down on your smoking, if not give it up altogether.
That's some great information. So thinking more specifically about dry AMD, you've mentioned that we've talked about this term geographic atrophy. Could you tell us a little bit about what that means, what that is?
Sure. So those with dry macular degeneration who have more significant vision loss and more significant visual disability generally have what we call geographic atrophy. And what happens in geographic atrophy is that those tissues, the layer underneath the retina in particular, which is called the retinal pigment epithelium. And remember, this is in the central portion of the retina. So this is in the portion of the retina that you use to read with and to see colors with and to drive with. We know that those layers underneath the central portion of the retina can essentially wear out. And the photoreceptors that overlie it, so the rods and the cones, and in the center it's mostly cones, rely on that layer for its nutrition and for its metabolism. So those photoreceptors die and people end up with these blank spots in their vision. So often those blank spots, surround the center or they're close to the center. And as it progresses, it can involve the center and lead to substantial vision loss in the center.
So does everyone who has dry AMD, will they have geographic atrophy as well, or will they develop it eventually?
No, absolutely not. I mean, there are lots of, the vast majority of people with dry macular degeneration will have some vision loss, but not substantial vision loss. But certainly there's a subset of patients with dry macular degeneration who will develop geographic atrophy. When we think about when we kind of divide the proportions of patients that have dry macular degeneration versus wet macular degeneration, we usually say that about 85% of macular degeneration patients have dry macular degeneration, but 85% of the severe vision loss in macular degeneration occurs because of the wet form. But that's not 100%. So there is a small but significant proportion of patients with dry macular degeneration who can develop significant vision loss, and that's from geographic atrophy.
So you talked a little bit about some risk factors, primarily smoking, diet. Are there any other risk factors? Who's at risk for developing geographic atrophy and dry AMD?
I mean, certainly for dry AMD and geographic atrophy, but for all macular degeneration, the most important risk factor is age. So unfortunately, there's not that much we can do to prevent the march of time, but age is certainly the most significant risk factor. But there's some genes that have been found that put you at higher risk for vision loss from macular degeneration and more severe forms of macular degeneration. So things like If you have, close family members that have had significant vision loss from macular degeneration, that puts you at some risk. It's not a one-for-one correlation, but certainly a family history puts you at some risk.
For sure. And right now, what sort of treatments are there treatments available for dry AMD and geographic atrophy?
We don't have any approved treatments at the present time for geographic atrophy. There's a lot of exciting things in the pipeline. I think the most the agent that we will have the soonest is a drug from Appelis, one of our sponsors. So we know that what happens in geographic atrophy is that the immune system, the immune system has gone awry and causes damage to the central retina. So there are some molecules, some drugs that have been developed to block certain aspects of that immune system and hopefully slow the progression of dry macular degeneration. So there's a drug that will be coming. It has not yet been approved in Canada, nor has it been approved yet in the United States, but probably I think they anticipate approval pretty soon. But it's a drug that's given by intravitreal injection. That means it's a drug that's given by injection into the eye, and it's been shown to slow the progression of geographic atrophy. So for those patients that have geographic atrophy that hasn't yet involved the center, this has important implications because hopefully it can slow the progression of that atrophy so that the central portion, the very central portion of your retina can maintain vision. And then there are other more exciting, I think even more exciting things that are, I think, a little ways away, that are some years off, but things like stem cell therapy or cell replacement therapy, where those cells that have died hopefully can be replaced, and some vision, not only preventing the progression of vision loss, but hopefully restoring some vision that has been lost. And I think there are some exciting projects that are in the pipeline that are already in clinical trials that I think we'll see in the next, I don't have a crystal ball, but I like to think that within the next 5 to 10 years, we'll have, much improved treatments for geographic atrophy.
You anticipated my next question, but I noticed that in the question and answer, maybe I'll ask you this now, just while we're talking about treatments. We have someone named Terry who's saying that they'd like to hear more about the research on laser treatment to manage dry macular, that they're currently getting the treatment, but it's very pricey. Is this like a treatment you're familiar with? Would you think it'll be covered eventually by provincial health plans? Could you comment on that at all?
Yeah, I mean, there are some laser treatments that are being offered and that have been tried so that one of the markers of dry macular degeneration are these things called drusen. And when we look into the back of your eye, we see these little yellow spots that are called drusen. And we know that laser can be used to get rid of the drusen. Now, what sort of impact that has on preserving vision or reestablishing vision is not well known. There really isn't that good science to demonstrate the efficacy of laser for dry macular degeneration. So hopefully there'll be clinical trials that are done, properly designed clinical trials that will show that these are beneficial. And I think if those are done and if they prove to be beneficial, then I think a case can be made for those to be covered by our provincial insurance plans, but I'm not sure how soon that might happen. We really need the evidence before a case can be made to government to fund these treatments.
So I know, prior to the anti-VEGF injection treatments, laser was also used to treat wet AMD. Is that correct? Would this have been a similar type of treatment, or is this quite different?
This is different. I mean, the laser that was done for the wet form of macular degeneration was a pretty strong laser, and it was the goal of that laser was to destroy those abnormal blood vessels with laser, to ablate them with laser. This is a much lighter form of laser, much less destructive form of laser that we're talking about for dry macular degeneration. But because the original laser for wet macular degeneration was so destructive, it's kind of gone by the wayside. Haven't been doing that for a long time.
That's great. So if someone has, just another question on that, if someone has dry AMD, there's really, there would be no need for the injection, like an anti-VEGF injection, right? That is just purely when someone has develops into the wet form?
Yeah, the anti-VEGF, so the what Morgan is alluding to is a class of medication called anti-VEGF medication. So VEGF stands for vascular endothelial growth factor, which is a molecule that the body produces that promotes the growth of abnormal blood vessels and promotes leakage from abnormal blood vessels. And the way we treat the wet form of macular degeneration is we inject medicines into the eye that help to block the action of that molecule. So that doesn't seem to be an important player in the setting of dry macular degeneration. So we don't treat dry macular degeneration with anti-VEGF injections. So these antibodies that are directed at parts of the immune system that we know play a role in dry macular degeneration or geographic atrophy in particular are also given by injection, but it's an entirely different class of drugs. It's entirely different medication.
Right. So I think in the Q&A we had a question from Sue just saying what are we currently being given in quarterly eye shots and what's the difference? So I would assume that those quarterly eye shots are being given for wet macular degeneration, likely the anti-VEGF treatment.
Right. I mean, there can be some confusion. We sort of guide our treatment for the wet form of macular degeneration by disease activity. So the way we monitor disease activity is we look for whether those blood vessels are leaking. So we look for fluid under the retina or within the retina with some different imaging techniques that we have. So we talk about, the retina being dry, but the retina being dry in somebody with wet macular degeneration is different from dry macular degeneration. So I know it can be a bit confusing to our patients, but we want your retina to be dry, but it doesn't mean you have dry macular degeneration. It means you have inactive wet macular degeneration, and it's kind of an important distinction. And I know there's confusion out there, and I know it's easy to get confused, but they really are two distinct entities.
Yeah, there might need to be some language change there because the dry.
Yeah, we don't make it easy for you.
So you've talked a little bit about this, but just lastly, before we move on, how can people best manage symptoms that they're having of AMD or geographic atrophy? I know you've talked a little bit about things like stopping smoking, changing your diet. Is there other things that people can do?
Well, as we've said, those with dry macular degeneration certainly are at risk for the development of these abnormal blood vessels, for converting into wet macular degeneration. So the first piece of advice I would give is that if anyone has any significant changes in their vision, particularly if they see new black spots or blank spots, or if they have new distortion, they shouldn't sit around. They should try and get in to see their eye care professional as quickly as possible and try and ascertain whether they have the wet form of macular degeneration, for which we have a number of very good treatments. Because the earlier you get treated, and the sooner the disease can be arrested, the better the outcomes usually are. With respect to people with dry macular degeneration who are having difficulties with reading and color vision and that sort of thing, in this day and age, there are remarkable visual aids that are available. I mean, starting from something as simple as an iPad can really transform your life and make things that you are finding very difficult to do much easier. It won't make it as easy as it was before, but there are lots of ways you can continue and carry on doing the things that you like to do and want to do. And we talked about some lifestyle changes, about eating more fish, eating a diet rich in green leafy vegetables, the concoction of vitamins that are available to help prevent vision loss and progression in macular degeneration are all things you can do to help slow the progression of your disease.
Thank you so much, Dr. Kertes. Just one last question. When you were talking about, monitoring your vision, seeing if there's black spots or distortions, is that where someone would use that Amsler grid, that grid of lines?
Yeah. So what Morgan's talking about is a tool that we use and often give out to our patients, which is an Amsler grid. So it's essentially a graph, and it's a useful way to monitor for changes in your vision. So I don't think we're going to show one today, but an Amsler grid, the way we ask people to look at an Amsler grid is to do it with one eye at a time, as I mentioned before, and to look at the center of the grid. And if there are any lines in that graph that appear newly distorted, or if there are any blank spots in that grid that are new, then that's an indication to us that something may be changing and that you should get checked out. But certainly there are lots of other ways to look for distortion. If you cover one eye at a time and look at a door in your bedroom and see if it's got some kink in it or some bend in it, that's another. Or if you look at a hydro pole outside your bedroom window and see that it's not as straight as it ought to be, then that might be an indication that you need to get your eyes looked at and make sure everything's okay.
Great, thank you so much, Dr. Kertes. So we'll come back at the end. I know there's going to be lots of questions at the Q&A at the end. So thank you so much.
Good thing, stick around.
So now I'm going to turn it over to Dr. Larissa Moniz, who's our Director of Research and Mission Programs. And Larissa is going to be speaking first with Dr. Levi Blazer.
Thank you, Morgan. And thank you, Dr. Kertes. That was a really great introduction to dry AMD and wet AMD. And I think it will be a nice segue into Dr. Blazer's part of the session. So back in January, we announced the winners of our 2021 research grant competition. And we were really excited to award a grant to Dr. Sukhdev Sidhu and his team, and they're developing a new potential treatment for vascular retinal degeneration, such as age-related macular degeneration and diabetic retinopathy. So we're really excited to have Dr. Levi Blazer, who's a member of Dr. Sidhu's team, join us, and he's going to tell us a little bit more about the research and the impact it might have for eye disease, but specifically for AMD. So welcome, Dr. Blazer.
Thank you. It's good to be here.
So I was wondering to start off with, if you could just give us a little bit of information about yourself and the type of research you do.
Yeah, absolutely. So I was trained originally as a molecular pharmacologist. I got my degree at the University of Michigan, and then transitioned into industrial research in the pharmaceutical industry. Life brought me to Canada, thankfully, and I joined the laboratory of Dr. Sidhu at the University of Toronto. Specifically, I'm in a subsection of his lab called the Toronto Recombinant Antibody Center. And what we do, we call it TRACK, that's our acronym for it. What we do at TRACK is we have a system to develop fully human recombinant monoclonal antibodies. And we have a very large repertoire of these antibodies, almost 20,000 different unique molecules at this point, that target many, many different cell surface markers or pathogens or what have you, that we try to translate into some form of clinical value eventually. Something that is useful either in the clinic, or a lot of our work also is very valuable as basic science research agents as well.
So you mentioned antibodies, and so I think a lot of people, when they think of antibodies, they might think of their immune system creating antibodies. I was wondering if you could maybe just very briefly explain what an antibody is and why you would use that to create new treatments.
Yeah, that's a very good question. I think the concept of monoclonal antibody therapies really got a lot of worldwide recognition during this pandemic. I think a lot of people who may not have appreciated what antibodies can do started to see that with the monoclonal antibodies that have been generated against SARS-CoV-2. And essentially what an antibody is like you mentioned, Larissa, it's a class of proteins that are made by our immune systems. Antibodies generally are shaped kind of like a Y, where you have a stem and then two arms. And the purpose of an antibody is to go around your body identify things that shouldn't be there, things that are foreign, bacteria, viruses, cells that have grown malignant, etc. And what they do is, with the tips of these two arms, they can bind to that bacteria, to that virus, what have you, and direct the immune cell or the immune system to come in and clean up that foreign object. So that's kind of the role. There's a fair few complications and additional like immune complexities, but that's kind of the general idea of what they do in our body. Now, for us and for antibody scientists who are trying to develop therapies against antibodies, it's a little bit of a different story, because a lot of the things that we want to do may not be immune directed at all. So, for example, the molecules that we're making, we actually remove the ability, or we've engineered out the ability of our molecules to interact with the immune system at all. And instead, what we do is we make antibodies that bind to very specific cell surface receptors that are important for the maintenance of the blood retinal barrier. And they can activate the cells in that portion of the retina to grow and to form solid, tight junctions. So there are an awful lot of different things that one can do with antibodies besides just use it to direct the immune system to clean up something or to treat something.
So if I understand, you're sort of using the sort of intrinsic ability of an antibody to recognize something very closely and using that almost as a tool and to do other therapeutic purposes.
That is very much what we do. We develop antibodies because they are exquisitely specific at recognizing fully folded cell surface receptor proteins or other proteins throughout the blood or through the body, in a way that many small molecule drugs, like many current drugs out there, are just not unable to have the specificity of an antibody molecule.
Oh, that was actually, I was going to ask you this later on, but why would you use an antibody versus, say, a normal drug? You're basically, because they can recognize things better, they can bind a bit tighter, hopefully.
Yeah, I mean, there are a variety of different reasons why one might want to use an antibody. One, they're highly engineerable, at least the ones that we work with are, and that provides an awful lot of benefit. Another reason that antibody drugs can be beneficial over some of the small molecule drugs is that you can very finely tune the half-life of that molecule. So you may take baby aspirin during the day for cardiovascular reasons. You take one a day, right, every day. And that's good and that works. Most antibody drugs, though, they have a half-life. That means they can survive in the blood for about 2 weeks. So you can get an injection and it can last for a long period of time, a much longer period of time than an oral drug might last in the body. So that's one reason why they can be valuable, is you may be able to dose less frequently. Mm-hmm. Another thing is you can have much increased specificity or selectivity over certain drug targets that may be very, very similar in the body. So a lot of our drug targets that we try to develop therapies targeting, they may have 3 or 4 very closely related systems or other proteins in the body. And a small molecule drug, it can be very difficult to sometimes tease apart selectivity so that you only hit the one that you want and don't get some off-target effects, as we would call. And those off-target effects can often lead to side effects or unanticipated problems with those therapies. Antibodies, we believe, and we've been able to show, can have much greater specificity.
That's great, thank you. I've learned so much from that answer. We got a little off track, but I thought that was fascinating. No, that was so interesting. So you mentioned earlier in one of your answers about the blood retinal barrier and how the antibody for this project that you're working on is trying to improve it or something. Can you maybe describe what the blood retinal barrier is and why it's important in diseases like AMD?
Sure, absolutely. So in retinopathies, the blood-retinal barrier can be weakened. And this is I guess I should step back. The blood-retinal barrier is a system that protects your eye from the general circulating blood supply. So the eye is a very special organ in that you don't want to just have full exposure of your eye and the interior of your eye to everything that's in your blood. There are very sensitive neurons in there. There are very sensitive photoreceptors. There are lots of very sensitive cells in your eye. And so biology has devised a way to protect these sensitive cells by generating a barrier around the vasculature in the eye. And essentially, it's a series of cells that are very tightly connected. And these cells only allow certain things to pass through. They tightly control the flow of water through so that the eye pressure is maintained at the correct level. And they very carefully regulate what solutes, what salts and nutrients and such can penetrate into the eye. It's also very important. And like I had mentioned, in retinopathies, it is not uncommon for that blood-retinal barrier to break down, and that can lead to a variety of problems and damage to some of those very sensitive photoreceptors and other components of the retina.
And so that blood-retinal barrier, one of the side effects of wet AMD in particular is that the blood-retinal barrier breaks down. So I was wondering if you could tell us a little bit about how the new treatment might work to fix the blood-retinal barrier and how you sort of came to devise this new treatment. Sure. So we came to devise this treatment because we were very interested in a class of receptors, cells that are, proteins that exist on the surface of many of our cells. And these receptors fall into a family called the frizzled family. It's a little bit of a silly name, comes from the world of fruit fly research a long time ago, but we call them frizzled receptors. And frizzled receptors are very important growth factor receptors in the body. So they drive development through embryogenesis all the way through adult tissue. They are very important for stem cell maintenance, especially in the intestine. And they are very important for a lot of growth in our tissues. And for a very long time, people had been trying to develop small molecule drugs that would target frizzled receptors and inhibit their activity, generally for cancer indications, for oncology. But it really took antibodies to be able to get there. The small molecule frizzled antagonists were never quite specific. We've been able to generate antibodies that very specifically target every single member of this frizzled family. And whenever this frizzled receptor wants to signal and start driving growth of in a particular cell, it needs to not just have itself, but it needs to have a partner with it, a buddy, we call a co-receptor. And those two molecules, that frizzled receptor and its co-receptor, have to come in very close proximity to be able to generate the signaling inside the cell that's required to get it to grow and to form the tight junctions that are required for proper blood-retinal barrier formation. And so what we did was we had a large repertoire of antibodies that were very specific for each member of the frizzled family. And we had a large number of antibodies that were very specific for the co-receptor. And we started recombining them in a variety of different ways, what we call bispecific or multi-specific antibodies that don't just recognize one thing, but can recognize two or more different things. And through some from my end, some very interesting antibody science, we were able to show that a very specific arrangement where we have the ability to engage two of these frizzled receptors and two of their co-receptors on the same molecule, bring them all together into one cluster on a cell, we can very potently activate that signaling system in that cell.
And so by doing that, we were able to generate what the first set of frizzled agonists that were fully antibody based. And we've been doing some very interesting science with them. One of the most exciting things that we've been doing is we have generated a molecule that's very specific for a receptor that is used by the blood retinal barrier endothelial cells to grow and drive these tight junction formations. And we've been starting to study that in a variety of preclinical models, and it's been looking very promising so far. We're very grateful that you and the foundation were able to provide us with some support to be able to do some more of these preclinical molecules, preclinical studies to see how efficacious that this treatment could be.
So that's really interesting. So basically, these molecules are actually activating the pathway and causing them to basically recreate the blood retinal barrier that has been disrupted in AMD or diabetic retinopathies. Is that the case?
That is absolutely the case. And we could compare that to the VEGF inhibitors that Dr. Kertes mentioned. Those are actually the VEGF molecules that he mentions, or VEGF targeting molecules, are also antibodies generally, or antibody fragments that inhibit the activity of VEGF. They can bind to it and just keep it from binding to its receptor and activating that signaling system. And in that case, the VEGF activation can lead to increased leakiness of the vessels. So we're both kind of coming at it from this, the VEGF inhibitors and our molecules are kind of coming at it from different approaches, but trying to reach the same end, where we either activate or inhibit a signaling system to try and improve the integrity or restore the integrity of the blood retinal barrier.
So I know for this project, what the goal is to test out this new treatment in different animal models, and you're also going to compare it to anti-VEGF. So ultimately, do you see this treatment being a standalone treatment, or do you see it being used at the same time as anti-VEGF? Like, what impact do you think it would have for individuals who have AMD?
Well, they are very different signaling systems. So I think a combinations trial would be very interesting. I would love to be able to test that and see how they work in parallel. Because you're right. Right now, we've been comparing all of our work so far has been in comparison with VEGF inhibitors to see kind of how our system, which is it's a very different mechanism, stacks up against what is currently available.
That's great. So just before I let you go, you mentioned the frizzled receptors and all the work you've done with them. Are you also looking at either the same molecule or different molecules for other diseases, or is it specific for the eye right now?
We started with the eye because it was a very interesting place for us to start, and we had a good foothold with some of our collaborators. I mean, that's one of the things with science is we tend to get so specialized that you have to have a lot of good collaborators, and we're very fortunate to do that. So we started down the eye path because we had very good collaborators in place for that, but we absolutely are pursuing other frizzled co-receptor combinations for other disease classes as well.
Yeah. That'd be really interesting, because I know that anti-VEGFs, they came from a cancer treatment originally, Avastin, which has now moved into the eye. So it'd be interesting if it went the other way. Thank you so much, Dr. Blazer. So we'll have you back again in a few minutes to answer some questions at the Q&A, but I really enjoyed talking to you.
Great, thank you so much.
No problem. And Dr. Hurley, if you are around, if you want to pop your camera on. Hello, how are you doing?
Outstanding. How are you?
Good, thank you. So Dr. Hurley joined us sort of at the end of last year for a session, so we're so happy to have you back again. We thought you gave such good information that we sort of called on your specialty again. So thank you for joining us again.
My pleasure, absolutely.
So sort of the same starting question as we asked Dr. Kertes and Dr. Blazer. Could you just quickly introduce yourself and your work, and maybe provide a little bit of background about your work specifically with AMD?
Sure. So like Dr. Kertes, one of my great mentors. I'm a vitreoretinal surgeon here in Ottawa. So I'm part of the University of Ottawa Eye Institute and work at the Ottawa Hospital treating patients with surgical and medical retina conditions. And as an educator, I was the program director here for many years, training the young physicians, and currently serve as the fellowship director, training the more specialized subspecialty physicians within ophthalmology.
Thank you. And so we actually asked you specifically to talk about a topic called biosimilars, or a category of drugs called biosimilars, because they are relatively new to the eye space and relatively new in general in Canada. So we thought it would be really interesting to get a bit of background about them. So starting off with a really simple question, or maybe not so simple, what are biosimilars?
Perfect. You know, it's becoming a hotter and hotter topic within ophthalmology. It's something that when I went through medical school, this is not a subject that was discussed. So it's novel, it's recent, and it represents an evolution, I would say, within medicine in general. And so if you think of biosimilars, we first have to maybe go back a step to think of more simplistic drugs. So common drugs that we take, for example, aspirin or Tylenol, are small and relatively simple molecules that can be easily synthesized within an industrial plant by combining the right chemical ingredients together, and you can put them together and you can come up with a simple drug like, say, Tylenol. Tylenol's generic name, of course, is acetaminophen. And so another manufacturing group can take those exact same chemicals and so sort of copy the recipe and produce an equivalent, not an equivalent, but an exact replica of that simple drug. And so we call those generic drugs. And so I think the general population is very familiar with the term generics. And so if you have one of these simple drug compounds, and then an alternative producer is manufacturing an exact replica of it, we call that a generic. And so we've been familiar and we've been using that term for many years. When biologics came along, so the new category of medications known as biologics, biologics are much more complex molecules than the simple drugs that we've been using for a long time. So the biologic molecules are large, complex, huge compared to those simple drugs. And they can't simply be created by putting together or combining in a traditional manufacturing sense all of the elements. They take an organic system to actually produce them. And so we don't really manufacture them the way we did with the simple compound drugs, yet we'll use sort of a biological system. The best example, say, would be insulin as a biologic. It's produced by the pancreas. And of course, we use the pancreas of animals to produce insulin for humans for many, many years. Now we have better ways of making it, but we're still relying on cells to synthesize this much more complex molecule. Now that we have these much more complex molecules in ophthalmology, examples were given before of the anti-VEGF medications, terms that people may know like Eylea and Lucentis and Avastin. Now the traditional manufacturers of those, our manufacturing capability has been expanded with other manufacturers producing those molecules similarly by using a biologic system to produce them. But because the biological system is not exactly the same as the one that's making the traditional, let's say, Avastin or Lucentis, then the compound that produced may not exactly have every single atom lined up the same. So over tens of thousands of atoms, there may be a couple differences, but yet the effect, the desired effect on the end organ, so the desired effect within the eye when it's given, needs to be the same as the original biologic drug. So a biosimilar is an alternative to the original biologic drug in the way that a generic is a similar to the original more simplified drug that we're used to dealing with. So biosimilars are copies of biologics that are very similar but not identical to them, but should have the same effect when used within medicine.
I think that was a really great explanation. There was a lot in there. I liked your analogy of when you mentioned about a recipe. It made me think if you give two people the ingredients and the instructions to make a cake, they'll be similar, but they probably won't be exactly the same. They're probably both very tasty, but maybe not exactly the same. And I guess in a way it's...
Yeah. And I might even elevate that analogy a little bit higher to say, well, it's like baking a cake. If you really had a simple cake and you could really exactly copy it, but think of a more intricate something like, say, a rose. And so when you grow a rose, it grows out of the ground, and it depends on humidity, it depends on sunlight, it depends on temperature, exactly how that rose will form. So the way it forms, somebody else might grow a rose, it'll look like the rose, but it will be different, particularly if the manufacturing processes had some differences like temperature or humidity. It may look quite a bit different. And so reproducing a rose identically is much more difficult because it's a biological system than, say, reproducing a cake. And so it just really defines that we're dealing with almost large, nearly living things that interact with our body compared to more simple drugs.
So you've already mentioned some of the drugs that this could be related to, so the anti-VEGFs. So in this session, we're talking specifically about AMD. So I'm assuming that there might at some point be biosimilars for some anti-VEGF drugs. Are there any other eye conditions where biosimilars might be coming into play?
Yeah, so as a retina specialist, my first exposure to it was treating patients with uveitis. So patients who have an autoimmune disease where their own immune system is attacking their eye because of a deviation in the immune response. Biologics were very effective in rheumatology, in the specialty of medicine that treats autoimmune disease, for example. And so when we have an autoimmune disease in our eye, we sometimes give patients systemic medications to treat that autoimmune disease. And so things like Humira come to mind. Now, biologic versions of those medications have been available and used outside of ophthalmology quite a bit, but within ophthalmology, within certain subspecialties like treating uveitis. So that's where I first was exposed to this whole concept of biologics and biosimilars within ophthalmology.
Okay, so if we could maybe talk a little bit about the potential for biosimilars to treat some of these vascular retinopathies like AMD or diabetic retinopathy. So how you've described very well about biosimilars. Is there anything else you want to add about how a biosimilar for an anti-VEGF would be different than pre-existing drugs that are currently used to treat AMD?
Yeah, so that's what makes this topic so exciting right now, is the fact that they're starting to see biosimilars appear for the common anti-VEGF drugs that we use for Avastin, for Lucentis, for Eylea. And so I've been involved in the clinical trials of the biosimilars for Lucentis and the biosimilars for Eylea and looked at those results. And so the way that the original product drugs, the original anti-VEGF drugs that were specifically designed for the eye, they underwent a lot of clinical testing to ensure that these were effective and safe in the eye. And we really characterized how those drugs interacted and worked within the human eye. As biosimilars are introduced, really a lot of the work is done to show that the actual molecule will work the same as the other molecule in preclinical data. And then small clinical trials are released to show that, yes, it does work within the human eye. So the number of subjects involved, the size of the sample that's taken is very much smaller, but these drugs undergo a lot of work initially to prove in the background that their chemical structure is sufficiently close to that of the original drug that we expect them to have the same effect.
And so you mentioned you're part of some clinical trials. So are these drugs already available in Canada?
They are so the first ones are about to be approved for clinical use in Canada. They have been used within the context of clinical studies to study their use. So yes, they are being used now in Canada, but not quite yet available on a day-to-day basis to treat patients outside of clinical trials.
Okay, and so do you think there might be some on the market this year or next year? So it's in a relatively short time frame?
Yes. The first ones we'll see will be for Lucentis and the biosimilars to Lucentis, and they should be in clinical use this year. And that'll be followed by the ones for Eylea in the coming year or two.
Okay, so how do you think the introduction of biosimilars will impact an individual who is maybe getting anti-VEGF drugs, and how will that impact your practice, I guess, when you're talking to patients?
So if we look at why biosimilars are produced and made, again, we'll go back to the analogy with generic medications. They offer a less costly alternative. And so the agencies that fund healthcare in Canada and pay for us to use these expensive medications to treat eye diseases, and these biosimilars will be available for the major blinding conditions that affect the eye, diabetic retinopathy, macular degeneration, and any other diseases that we use anti-VEGFs to treat, for example, retinal vascular occlusions, and talked about uveitis, and neovascular glaucoma, and other ones before. So there's a large market and a large potential there for some economic savings if we introduce a product that is sold at a less expensive cost. So you can see there's a large motivation from the individuals that fund healthcare to consider these as a cost saving in medicine. And realistically, if you look at that and say, okay, as long as that money is not taken out of the treatment of eye disease or not taken out of medicine, but we use those funds to sort of pay for maybe additional research or allow the treatment of other conditions, expand our ability to treat more patients, you could see where this would be very beneficial to the system as a whole. But we also remember that we treat the individual patient that is sitting right in front of us. And patients who have been doing well on an existing therapy are now confronted with a new drug that has less clinical experience, and there could be some real concern on behalf of those patients or those physicians as to how it will affect their own individual patients, particularly ones that are treated with a different medication now and new alternative medications are being introduced. But the theory, the concept as a whole of having more choice in our treatment to treat patients and different drugs of different costs to treat patients is very appealing.
Yeah, that's, I think, two really good points about obviously the potential cost savings there could be for the system as a whole. But then when we're talking about individuals, we are treated as individuals. We're not treated as a whole. So how would you sort of navigate that with your patient? I guess you've already dealt with this with uveitis. So if, say, somebody switches over to a biosimilar, are they allowed to switch back to the other drug if it isn't working? Do you have any experience with that in your other experience?
So biosimilars have been rolled out in different provinces. We haven't seen here, I operate and treat patients within the province of Ontario, but also a lot of patients from Quebec. And we don't know exactly how these will be implemented. In reality, it comes down to me being able to provide the medicine to my patients. And to do that in our system requires me working within the provincially funded healthcare system. And so I rely on those people who make decisions on which drugs will be paid for to help me offer free and inexpensive treatment to the patients versus a drug that may not be provincially funded and may be very costly to the patients. So we have situations where suddenly biologics aren't funded and only their biosimilars are. Then we have to have a very detailed discussion with our patients about the choices they would have and the relative cost of each of those. But really at this time, we don't know how these drugs exactly will be rolled out within this province in terms of how it might change which medications are paid for and which aren't.
Okay, so I guess that's a good point, sort of a little bit of a wait and see. So as an organization that is really all about patient choice, we obviously are both very interested to have biologics here, but also, or biosimilars, but also really keen that patients have the treatment that works best for them. So sort of,
very well said.
Yeah, continuing work to come. So yeah, you did mention some examples of biosimilars for other eye diseases. Do you have any other either experience or just you've heard about how biosimilars have potentially been used in other diseases and what the impact has been? Positive, negative, anything?
Yeah, we can think of simple diseases like treating the type 1, type 2 diabetes on patients who require insulin, and now we have biosimilars for that. Their largest use, as I alluded to before, are probably in some of the cancer therapies and some of the rheumatologic conditions, the autoimmune conditions, where again, they've provided some cost-saving alternatives, where that funding has then been used to continue research, to treat more patients, for example. But there has been specifications made within those fields to say, well, if a patient isn't responding as well as they should to one drug, you still have the ability to try another drug. So it's wonderful then to have different drugs that we can try, because patients are individuals and each individual may respond differently to one medication or another.
This was a really interesting discussion, Dr. Hurley. Thank you. It's given me a lot more to think about with biosimilars. We've definitely been hearing more about them as we know that some are coming to market. So I'm glad that we got to start this discussion. I know we'll be having more as they actually are approved.
Absolutely. It's an exciting time in our field whenever we have the ability to have new medications, and retina has really benefited from that in the treatment of retinal disease over the years, and glaucoma, so many eye diseases. Just the number of choices we have for medications and surgical intervention continues to explode.
Thank you. Morgan, I think you can come back. Yes.
That was so great. I have to say that is definitely the best explanation I've had so far of biosimilars. I'm always a bit confused, and that was really, really helpful. Thank you so much. So thank you to all three of our speakers. I'm going to give everyone just a moment to take a small break. And if you have questions, I notice we have a lot of questions coming in, which is fantastic. Please feel free to send in some more. I am just going to hopefully get my colleague Susan on screen here. I can just give that a second for Zoom to let her in, to talk to us a little bit about an exciting event that's coming up with Fighting Blindness Canada. Susan, are you there?
I am here, just waiting on, oh, hang on one second. There we go. There we are. Hi.
Hi.
There we go. Technology, you know, feels like a Monday.
Absolutely. Short week, but
It is. Well, thank you very much, Morgan. I appreciate the time, and it's been a fascinating webinar. So thank you, everybody, very much. And you know, we at Fighting Blindness Canada rely on the support from our wonderful community, like all of you. And with your donations, you are impacting vision research and the fight to end blindness and all the work that our doctors do. We offer a lot of ways that you can become involved and share in the fight and participate in any of our fundraising events. You can find a lot of information under the fundraising events section on the FBC website. And a couple that I want to tell you about today is, first up is our Screens Off for Sight Challenge. That's happening on March 19th. And with the pandemic, the last couple of years, a lot of us have increased our screen time, me included, which leads to eye fatigue, additional redness, watering eyes, and it's just not great for our eyes. So we are challenging you to turn off your screens for 24 hours, starting at 6 p.m. on March 19th. Give your eyes a break and fundraise for vision research at the same time. Challenge your family to do it with you. Register and create your fundraising page, and then ask for donations for every hour your screen is off. Personally, I'm asking my friends to donate $1 for every hour, $24, and I plan to get outside to walk my dog, and then do a little painting and just do things off the screens for that day. You can get more information and register at fbcscreensoff.ca and create your own fundraising page and join us and see if you can do the challenge. Then after that, this June, Cycle for Sight will be coming back, and we are so excited to come back this year, and we are aiming to come back in person. And our in-person cycling events are going to return to the Toronto area in Alliston, Ontario. We'll also be in Langley, BC, and St. John's, Newfoundland. It's a great one-day ride if you haven't joined us before. It's safe, there's scenic routes, and there's various distances that you can choose to ride and cycle. And then, of course, there's the celebration at the finish line. But if you don't want to cycle, we're also offering tons of virtual activities to just move with us and be active. And together, let's move. Let's move vision research forward. Let's move together and find treatments and cures for vision loss. So again, all you have to do is visit our cycleforsight.ca page, tell others about your dedication to finding cures and treatments, and then ask for their help by donating to your personal fundraising page. FBC will take care of all the rest with tax receipts and thank you info to all of your donors. You just have to share the word and ask for donations to a cause that you believe in. Our team's here to help you with your fundraising, whether for Screens Off for Sight, Cycle for Sight, or any other personal fundraising that you would like to do to help us move our mission forward. We're here for you at any time. You can contact us at info@fightingblindness.ca. And of course, all this information is included in FBC's monthly e-newsletter, so be sure you've signed up for that as well. And I'm sure there'll be a link for you in the follow-up information. Thanks again for your time and for joining us today. And I hope to see you participating in one of our fundraising events very soon. Thanks.
Awesome. Thank you so much, Susan. Thank you for joining us today. And we're looking forward to these in-person events coming back for sure. Exactly. It's about time. All right. So I'm just going to have a look now. We're going to get into the questions. Okay. So let's see here. So if I could ask our esteemed panel to turn their cameras back on and join us, that would be great. All right, so let's see here. So we've got lots of questions that have come in, which is fantastic. So we have a question for Angela, and she says, my mother had macular degeneration and she was a heavy smoker, but I've never smoked. Is there a chance I could still inherit this disease, and what should I be watching out for? Maybe I could go to Dr. Kertes for that one.
Yeah, for sure. I mean, in truth, everyone is at risk for macular degeneration. There are certainly people who are at greater risk, and there are lifestyle factors that put you at even greater risk. So, I mean, the fact that your mother had significant macular degeneration doesn't mean necessarily that you will. Certainly, the fact that you're a non-smoker is in your favor, but just because you don't smoke doesn't mean you won't get it. I mean, it decreases the likelihood, and it may delay the onset of it, but and I would encourage you not to start smoking, but it doesn't mean that you're immune to getting macular degeneration.
It's all about risk management.
Mm-hmm.
We have a bunch of questions from various people here, both in the chat and the Q&A, and also people who'd sent in questions before about vitamins. I mean, we always get lots of questions about vitamins when we talk about AMD. So people are asking, what kind of vitamins do you suggest, or can you name the vitamins? I know there can be various types. Dr. Kertes, do you want to answer that one as well?
Sure. I mean, what we know about, the prevention of vision loss from macular degeneration, vitamins, stems largely from a large clinical trial that was done, that was sponsored by the National Institute of Health, called the Age-Related Eye Disease Study, or AREDS. And there was AREDS 1 and AREDS 2. So so some of the vitamins that you will see that are purported to help prevent vision loss from macular degeneration will have the label AREDS 2 on it. So there are different manufacturers. I think there's even a Costco brand. So and I usually encourage people to get whatever is on sale. But the ingredients, so they have some lutein in them. People are often have heard about lutein and know about the beneficial effects of lutein, but it's more than lutein. There are other antioxidants. There's vitamin C, vitamin E, there's zinc. So there's a concoction of, you have to look at the ingredients if it doesn't tell you that it's AREDS 2. And that same concoction of vitamins in whatever formulation you find them in are what have shown to be beneficial in a large randomized clinical trial.
So we have another question on the same topic of vitamins from Catherine, and she says that her mother has dry AMD and has vision loss associated with that. And she's wondering if she should be taking AREDS2 vitamins as a preventative measure. Would taking those vitamins help her prevent getting AMD?
I mean, that's a very good question, and it's a question I hear pretty often. I used to hear it more often before COVID when parents were allowed to come in with their children for their appointments, and the son or daughter that had brought their mother or father in for their appointment would ask me, my mother has this terrible problem, should I be taking vitamins? So we only recommend that people take those vitamins if they have, what we would consider as high risk dry macular degeneration. And the best way to ascertain whether or not you have high risk dry macular degeneration is have someone to look into your eye and look at your macula and see if you have features that we think put you at higher risk for the development of abnormal blood vessels or significant geographic atrophy. So just because one of your parents has it doesn't mean that that doesn't necessarily in and of itself put you at high risk. Usually we define your risk profile by looking into your eye.
I have another question here for Dr. Kertes. So Christina's saying that 40 years ago, she lost most of her sight almost overnight. The doctors at the time were puzzled. They came up with macular degeneration, but she was not given treatment. The only treatment she had was drops for dry eyes and had some laser several years ago. And she's saying that her pressures are up now. And she's wondering if taking drops to lower her eye pressure will have any effect on her macular degeneration.
So high pressure in the eye is associated with another very common, very visually disabling disease called glaucoma. And if she was advised by her eye doctors to take drops to lower the pressure in her eye and prevent the progression of glaucoma, I would certainly encourage her to carry on and continue to do that. But there isn't really a connection between glaucoma and macular degeneration. So it's not likely to have an impact on her macular degeneration. I just want to point out, I'm not sure how old she is at the present time, but if she had the diagnosis of macular degeneration as a young person, there are many diseases that can affect the macula and impact the macula. They're not all AMD. Remember, the A in the acronym AMD stands for age-related. So there are other macular diseases that are not AMD that are distinct from AMD. They may end up looking very much like AMD down the road, but that's a different group of disorders.
Great, thank you. I'll give you a moment, Dr. Kertes. I'll give you a little break. I have a question from Gina for Dr. Blazer, and she's asking, I think you did touch on this a little bit in your talk, but so she's asking if this particular antibody treatment that you're developing could also hold hope for Stargardt disease or other inherited retinal diseases.
So possibly, yeah. I'm not. I can't speak specifically to Stargardt disease, but the impetus that we had for focusing our attention on the eye was due to a series of congenital inherited diseases like Norrie disease, Coates disease, that do have implications for this pathway. So there is some possibility.
Great, thank you. I think this question, maybe I'll give this one to Dr. Hurley. So David's asking, can someone please explain the difference between Eylea, Lucentis, and Avastin, and is one better than the others? And actually, this is a common question. I think we have it several times. People are maybe confused about what the differences between these drugs are.
That's an excellent question. So they represent the classic anti-VEGF medications that are used for wet macular degeneration, as well as a variety of other eye diseases that depend on the molecule VEGF to cause or allow the disease to progress. So we think of Avastin as sort of the original one. It was the one that was in clinical use before the other two. And it comes from the observation that when Avastin was being used to fight cancer in the body, it did so by stopping the growth and proliferation of new abnormal blood vessels, which is exactly what happens in macular degeneration. And so an initial foray into it by using Avastin on a systemic level to treat wet macular degeneration demonstrated that blood vessels in wet AMD could be suppressed with the use of that medication. But the problem was then you expose the whole body to the systemic side effects of this medication. And so it was determined, and quite fortuitously through the brilliant innovations of physicians in Miami years ago, that you could take a very small quantity of this medication, inject it into the human eye, and it didn't have any deleterious effects on the eye. It was quite safe, but it would still be able to suppress the wet macular degeneration and halt the growth of those new blood vessels. And so Avastin being already available to the world and to the ophthalmic community was really the one that was first grabbed on and used. But at the same time, you know, Lucentis was, well, before that even, Macugen, I should comment on, another anti-VEGF medication that we don't use today, was being developed concurrently using that concept of suppressing VEGF, but now designing a medication specifically to be used in the eye. Turns out that medicine wasn't as successful because it blocked only one element of the VEGF cascade. So along came Lucentis, which provided more what we call pan-VEGF blockade, so blocking the whole VEGF family, or at least one subset of the VEGF family, and was much more effective at stopping the progression of wet AMD. Avastin basically blocks the same components of the VEGF pathway as Avastin. So Lucentis and Avastin are very, very similar. The difference being one is formulated, designed specifically to be used within the human eye and is a smaller compound. The other is designed to be used systemically, so it has features in it which allow it to stay within the circulation, the systemic circulation, longer. That's Avastin. That's very desirable if you're treating a cancer, a systemic cancer. But of course, we want our molecules to stay in the eye and not live in the human body outside the eye long, because we don't want any systemic effects when we inject something into the eye. So Lucentis designed to use and stay in the eye compared to Avastin. Eylea was another molecule designed. It's newer. It was released later than Lucentis, and it binds a little bit more than VEGF, and so it was felt that binding some other of the cytokines that are implicated in the wet macular degeneration pathway may provide some additional benefit. So we have the three drugs. The fundamental difference when Eylea was used in clinical studies was that it was dosed every 2 months, and Lucentis in its original study was dosed every month. So people thought, well, perhaps we'll get longer duration, longer treatment intervals, longer interval between treatment by using Eylea compared to Lucentis or Avastin. But it turns out that Lucentis and Avastin can actually be dosed at longer intervals too. And that dosing time is very specific to each patient. It seems that each patient requires a different frequency of injection. So it's really very specific on the patient and less so on the drug. And so those drugs, one represents an earlier drug, and second one, Lucentis, that we've had more experience with, but Eylea has been with us now for a long time as well. There's studies that came out, some major eye studies that looked at the difference between the drugs, and the basic conclusion was they can all be effectively used to treat conditions that are based on VEGF. And now we're seeing the next generation of anti-VEGF molecules coming along. We have Beovu or brolucizumab, we have faricimab, and so we're seeing now another generation of these medications available in Canada.
So, you know, I think if I'm hearing you correctly, Dr. Hurley, and I think a lot of people have these questions. You know, I've seen it popping up, a lot of people saying, I'm not responsive to this medication. Would this one be better? You know, this medication is more expensive. This one is cheaper. But from what I'm hearing from you, it's really based on the individual patient and kind of almost a little bit of trial and error to see what's going to work for the most amount of time between injections. Is that correct?
Yeah, it's interesting that a vial of all three of those medications basically costs almost exactly the same. The difference is the vial of Avastin, of course, can be split into multiple doses because the big vial is used to treat the whole systemic person. And as a result, the cost comes down to a fraction of what the other two retail for on an individual basis. But there's some advantages to having your compound delivered to you in a specific dosing ampule that was designed and tested and formulated to be safe, come right to you and go into the eye, whereas the Avastin has to be compounded within your pharmacy. And so there's huge cost savings, but it can bring in other, secondary issues that it serves to have a very long debate if we really wanted to go into all of the subtleties of the difference between the medications. But I would tell people to be confident that there's multiple medicines to choose from, that physicians who are making these decisions try to make the best one for their individual patient sitting in front of them. And we're lucky, again, to be able to have different medications to try in different people to see which one may work best for that person sitting in front of us.
Absolutely. I have another question here about biosimilars from Gregory, Dr. Hurley. So Gregory's asking, could slight molecular differences in the biosimilars create unwanted side effects?
So that's the big question, that we're having. Could it create unwanted side effects, or could it affect the wanted effects? Will they be as effective in suppressing the wet macular degeneration and improving the patient's vision? And so there are initial relatively small trials that do show that these drugs are as safe and as effective as the original biologic. However, sometimes real effects are only seen when much larger number of patients are exposed to and use these medications. And so when these medications are introduced, there'll be considerable post-introduction surveillance on those compounds to see if we're noting any of those changes to help guide us in our best decisions about which drugs to offer our patients.
Thank you. Yeah, that's really interesting. So I have a bunch of questions here, and I'm seeing a lot of chat in the chat box as well about genetic testing for AMD, and then maybe also the link between genetic testing and what vitamins that people should be taking. So are there recommendations around getting genetic testing for someone who maybe whose parents or siblings might have AMD? And what is there any sort of benefit to getting genetic testing, number one? And number two, does that have any sort of link to what vitamins you should be taking? Maybe I'll throw that to Dr. Kertes.
Yeah, that's a common question we receive, and I'm not sure we know the right answer to that. There is some evidence to suggest that people with certain mutations related to macular degeneration may, not just not have a beneficial effect with taking zinc supplements, but may have a deleterious effect with taking zinc. So that's an area of some controversy. The ARID study, which I alluded to earlier, didn't show any real link, didn't show any real genetic linkage to harm being done by those who took zinc for macular degeneration. Because there were so many people enrolled in that study, they would have thought that if that was a real effect, that it would have had some impact on the overall outcome of the ARID study. But having said that, there is some evidence, and it's published and it's available in the peer-reviewed literature that suggests that some people with some AMD-related mutations should avoid taking zinc supplements, and that in fact it may in fact be worse for their AMD than and not just not beneficial, but in fact worse. But I think the jury is still out on that, and it's still an area of some controversy. And there are lots of smart people who are invested in that discussion that don't agree. I'm not sure, Dr. Hurley, if you have anything to add to that, but I'm sure it's a question that comes up frequently for you as well.
Very common. And my answer is that we know, macular degeneration is clearly a genetic predisposed disease. We see large variations according to your race, your ethnic background, who your parents are. So there's a strong genetic component. Typically, the studies that suggest certain complement factors, certain genetic makeup may influence your response to some of the recommended ocular vitamin supplements to prevent the progression of AMD. A lot of that is based on a sub-analysis, which were not intended outcomes of some of these trials. And so they have to be taken with a grain of salt, because you're cutting your patient population in ways that the study wasn't designed to be balanced, and you could get spurious results. You could look and say, okay, everybody born on Tuesday versus Wednesday versus Thursday, and find that people born on a Thursday shouldn't take it. So I take with a bit of grain of salt when we do sub-analysis within a patient population in a large study that was designed for a different purpose. And that's where some of the evidence comes from to suggest certain people should or should not have these supplements. But I go back to the basic papers, the largest papers, I think the most relevant papers that show an overwhelming benefit for patients as a whole, and also show that this conferred not only benefits to their eyes, but to their overall health. So people on the supplements had less mortality and things like that. So my advice to patients is, it's under debate, it's being looked at, but until we have better evidence, rely on what I feel are the most reputable and less influenced studies. And I continue to strongly recommend ocular vitamin supplementation to all patients who would have fit into the category that benefit from it in the ARED studies.
Thank you. Thank you both. I have a question now for Dr. Blazer. So how soon do you think that your treatment would be tested in the clinical trials? And what would you need to see in your experiments before your treatment can progress to that stage?
Well, we are still very much in the preclinical stage of our work. So it will be at least a few years until we would be able to pursue clinical trials if our molecules are able to pass all of the requirements that are needed to get to that stage. So there's a lot of safety and toxicology studies that need to go into a package that is required to be allowed to do clinical studies, and also more efficacy studies, really figure out where the niche is for this particular molecule. So I would expect it will be a few years minimum until we would be able to know exactly whether or not it will be a viable candidate for a clinical trial.
Thank you. And another question for you. Are you studying any other treatments or developing any other treatments for eye diseases specifically?
Right now, we do have a number of VEGF inhibitors, but obviously those have been, there are very good options out there for VEGF inhibitors in the clinic. We do have our own that we are using in other indications and for other reasons. But the predominant eye project that we have right now is this frizzled agonist.
Great, thank you. I'm seeing a lot of questions here too about lutein specifically. Helen's asking if there's a correct dosage of lutein. We had another question about what sort of foods should people eat to increase lutein? Would either Dr. Kertes or Dr. Hurley like to comment on lutein specifically?
Sure, I'm happy to comment. So lutein was one of the ingredients in the ARED supplement. I believe the dose is 20 milligrams per day. You can correct me if I'm wrong, Bernie, but I think that's the correct dose, and that's the dose in the ARED supplement. But so I'm not sure if the question was whether or not people should megadose on lutein or not. The answer is not that we know of. And probably lutein, it can be argued, and it may be rational that lutein is the most important ingredient in those supplements, given the biochemical makeup of the in the macula itself. But remember, the studies that have showed benefit include lutein for sure, but include a handful of other vitamins and zinc as well, a handful of other antioxidants and zinc as well. So our advice is usually not to take lutein alone, but to take it together with the other antioxidants and zinc in the recommended doses.
Great, thank you. I have another question here from John. I think going back to potentially, I think these were for treatments for wet AMD specifically. And he says that he's heard that a reservoir can be inserted into the eye, and therefore he may not need to go for his quarterly injections. For wet AMD. Could someone share more about that particular option?
We look at the burden of treatment in AMD, and one of the major factors is, patients have to come very frequently for their intravitreal injections, and that places a burden on physicians who have to do that and running very busy practices. It puts a big burden on the patients and their families, especially in times like these in winter when it's difficult to make it to your visits when you get other illnesses. So there's lots of reasons why coming for very frequent injections is very difficult and costly to society, to patients, and to physicians in their offices. So, we're always looking at a way to space out how to the number of injections needed. And many ways have been proposed. There's been genetic therapies proposed where perhaps one injection into the eye using a viral vector on a gene may be able to give the eye the ability to produce its own anti-VEGF medications, or give the eye, turn off the production of VEGF in the eye. There's been the proposition that was mentioned by the individual who asked the question about these reservoirs that can be sewn into the sclera. And there's a couple that are out there and are being used. The most common one, can be refilled every 6 months and then slowly releases a version of Lucentis that's similar but not exactly the same as what we're using today. It's difficult, however, to reload those reservoirs. It's quite a tricky procedure. And when you insert that, it takes a surgical incision to place that into the human eye. And then you have an indwelling foreign object within the eye. And so you have complications of vitreous hemorrhage, retinal detachment during the insertion, and you have the concern of having that indwelling potential conduit for infection. So personally, I'm not sure that that's what we're going to see as the future, but it is being very actively studied. I think other ways of taking our existing drugs and making them last longer is being proposed. So reformulating our existing medications, so you get 4 times the dose in one injection, and therefore a much higher dose to begin with, so it'll decay a little bit longer before it reaches its subtherapeutic level, is being explored. And those next generation of anti-VEGF medications, which are here now and available to use in Canada, which again basically pack a lot more molecules into one dose. So it's still a simple injection, but they may have a better drawing effect on the retina and a longer duration of effect. And I think it's likely that solution that we're going to see as a way of prolonging our interval of injection rather than some of these indwelling devices. But I'll make that comment that that's a very personal opinion, having looked at the risk-benefit profiles of what looks like will be available in the near future.
Great, thank you so much to everyone today. I know we've had so many questions and there's so many that we're not going to have time to get to, but I really appreciate all of your answers, and also everyone out there for sending in all of those fantastic questions.